time of the year when classes are getting over and there's a lot of things going on, so it's great to see you all. Indeed, I will be talking about a problem related to the large-scale structure of the universe, but yeah, there are lots of interesting problems in astronomy, so if you're interested, you should talk to Moss. And also, I just will point out, we have for the American Statistical Association an astrostatistics interest group, and this year we are running for the first time, a student paper award in astrostatistics, and the winner will get a $500 cash prize. I mean, we're past the deadline this year. I'm saying this more for next year. If you're interested, just you can join the interest group. It's free for anyone really, and then you can submit a paper and perhaps win. And then you'll also present at the astrostatistics interest group topic contributed session at the joint statistical meeting. So anyway, if you want more details about that, you can talk to me later or email me. So what do I mean by spatially complex data? Here are two examples. This is an image that's a simulation, a cosmological simulation of the large-scale structure of the universe. The brighter regions are where there's more matter, and the darker regions are where there's less matter, or you might think of it as where there are voids or cosmological voids, so low-density regions. As I said, this is a simulation. It's actually a dark matter only simulation, but what's important to notice is the structure that's present. It's kind of a web-like, complicated collection of matter. And what astronomers might want to do is pull some information from this and maybe say something useful about the universe. So I'll explain a particular problem that I'll focus on. But so this is a very, very large scale, right? In many ways, the largest scales that we can even begin to conceive of. But if we also go down to very small scales, we have an example here of a human fibrin network. So fibrin is a structure that forms when you get a cut and your blood is clotting. So it's a protein structure. I don't know many details about fibrin, except that it also has this kind of web-like structure to it. That's kind of interesting, and what the researchers who work with fibrin might want to understand is how human fibrin differs from other species, other animals. But also you see that we have kind of places where the strands kind of overlap, and then what we might think of as lower density regions as well. And so in both these cases, we have spatially complex data. In this case, I want to point out that the data, this is a 2D image of a 3D simulation cube. And each point, each kind of colored region is actually a point. So we might call this a point cloud data set, while what we have here is image data. And so I'm going to talk about some methods that we could apply to the image data or to point cloud data and do statistical inference with that. So I'll begin with an introduction to persistent homology and discuss two popular ways for carrying out the computations for persistent homology. I'm going to then talk about a hypothesis testing setting where we might want to carry out hypothesis tests on these spatially complex data. I'll give the examples, of course, in a second. And then I'll have some concluding remarks. So that's the general outline that I will be following. So what is persistent homology? So persistent homology is often what people mean when they speak of topological data analysis. It's not uniformly what they mean. There are other areas within TDA, but it's a very common tool that's employed for topological data analyses. And it usually begins with some data set that you want to get some sort of topological information from. Then you get your topological summaries. These will be the persistence diagrams in our case here. And then with those summaries, then we want to do something with them. We might want to carry out some sort of inference, maybe hypothesis testing. Maybe you just want to visualize or understand some features in your data set, your complicated data set. Or you might want to do classification. Maybe you want to use these summaries as inputs into a classification model. So that's kind of the workflow that one might imagine with persistent homology. Okay, so the first example I'll talk about is for hypothesis tests for human versus monkey fibrin. So this particular study here looked at a number of different species fibrin, and what they tried to do was to see which species had, could they detect differences basically between the fibrin of these different species. Understanding that could be useful for things like developing animal models. So you're doing some sort of, maybe you're treating some sort of biomaterial to help blood clot better. What sort of animal would have the closest type of fibrin to humans, for example? But what they did is they randomly sampled 100 strains within each of these images, and then measured the thickness of those strands, and then carried out just very standard hypothesis tests, ANOVA type tests, on the sample of 100 strands from each of the images. And so that they can do that. They found certain differences. But what we're going to try to do is use this this human fibrin image and the monkey fibrin image, and carry out a hypothesis test to compare some sort of topological structure between them. So rather than just focusing on the thickness of the strands, we're going to try to say something about the topology of the strands.
So I have a stupid question. So so for the human, do you think everyone has the same fibrin topology? Just wondering.
That's not a stupid question. That's a great question. And in this case, so what we're going to find is we actually only had access to one human fibrin image and one monkey fibrin image. This is the only human fibrin sample I've ever seen. So I have no idea if this is actually representative. So what one would ideally want is a bunch of samples of human fibrin and a bunch of samples of monkey fibrin in order to have an appropriate hypothesis test. What we're going to actually end up having to do is cut this up and just say, this one human sample, how does it compare to this one monkey sample?
And this is a 2D image, right?
And it's a 2D image. Yeah.
Although this is actually a 3D structure.
Yeah, it is a 3D structure, but apparently they only look at these images as far as I could tell. But yeah, if we had the actual 3D image, that would be even better.
And the image is like a slice, or is it a projection? Is it a compressed 3D image?
I think it's a compressed 3D image, is what I would infer from their paper.
And I have a stupid question. What is a real stupid question? What is an oryx?
What is a? Oh! No, that's not a stupid question. Every time I show this image, I have to Google again what all these things are. And so, I mean, I know human, I know penguin, monkey, rabbit, sea turtle. Okay, so I think this is something that's similar to like an antelope or a deer. Yeah, and so. Yeah.
I mean, how did they come up with that one?
They had some justification. I'm not as familiar with all the different species and animal things, but yeah. Actually, I should maybe start putting notes on here too for all of them that are less familiar. All right. Any other good or stupid questions? All right, great.
That's the oryx.
And those were all good questions. Okay, so then the other example is with the universe data that I showed. I showed one realization of a cosmological simulation. In this case, we have two realizations of a dark matter only simulation that was run with two different settings, and the property that was changed had to do with something about the nature of dark matter. So we have a realization with cold dark matter and a realization with warm dark matter. We can just very simply understand it as cold dark matter, the particles move slower, and warm dark matter, the particles move faster. So there's this difference, and the general received theory in cosmology is that we have cold dark matter, but there's still interest in investigating differences in the properties of dark matter to see if there's maybe more going on, and just to better understand the nature of dark matter, the potential particle nature of dark matter. So in this case, what we're going to do, again, we only have one realization of each, and we want to test, we want to see if we can detect differences in the structure between the two. Now we can look at this and say, well, yeah, it looks like there are some differences. It kind of seems like they have similar large-scale structure. but can we actually detect smaller scale differences? So that's what we'll be focusing on with this example. Okay, so many of you may already be familiar with homology. I was not when I got started in this. It's not standard in a statistical education to learn about homology. So this is my quick summary of what we're looking for. And basically with homology, we're looking for holes. So we have zero-dimensional holes, which at least in statistics we would call those clusters, connected components. And then we have the first-dimensional homological features, which would be like loops. And then two-dimensional homological features would be like voids, or the interior of a soccer ball, or something like that. And so the Betty numbers are going to count the number of the different dimensional holes. So Betty 0 is going to count the number of connected components, the number of clusters. Betty 1 will count the number of loops, basically, and Betty 2 will count the number of voids. So this row has examples where we only have a single connected component, and the columns, in this column, we have no loops, and in this column, we have examples with one loop, and in this cell here, we have 2 loops, but only one connected component. Okay, so if we look in 3D, here we have an empty torus, and there's one connected component. There are actually 2 loops, right? So there's the loop that goes around this way, and then the loops that go around, the loop, I should say, that goes around kind of the interior. And then the inside is hollow, which is our H2 feature. Okay, so counting holes, that's easy enough to do when you have something like this loop, and maybe even if we had just randomly sampled on the loop, where in this case we have 15 points that were uniformly sampled around this structure. The homology of this would be Betty 0 would be 1, Betty 1 would be 1. But if we go over here and just directly think about the homology of it, we have essentially 15 connected components and no loop. But we see that there's a loop there, right? Like if someone gave you this data set, you would maybe even immediately think, oh, well, it looks like it's on a loop, sampled on a loop. So that would be easy enough. But then, what do you do when you get to a data set like this, where you have potentially millions of points on this complicated structure, and you want to estimate the homology of it, right? Suddenly it's not so straightforward. You don't want to sit there and be like, well, here's one loop, here's another, or you want to have someone else compute it for you, such as a computer. Okay, so persistent homology is going to give us a multi-scale version of homology, and I'll talk about how that's done with a particular filtration. But basically what it's going to help us to do, what persistent homology helps us to do, is to compute the homology of a particular data set across a particular filtration parameter. Okay, so how is that done? One way that's done is using what's referred to as a Ritz filtration. So again, we have our nice loop here, we have our 15 points, and we're just going to put a ball on the points and start growing the radius of the ball. And as the radius grows, eventually the balls start intersecting, and when that happens, we would say that the points become connected. So this is just one connected component. These started as a connected component, but at a very small radius, the balls intersected, and the 2 connected components merged into one. And so we could continue to grow these balls over the data set, and at some point they're all going to be connected. In this example, that doesn't always, at some point they're all connected, except there's a space between these 2. If we increase the radius a little bit, all of a sudden all these are connected and it forms a loop. So at the radius at which that happens, we would say that's the birth of a homological feature. So this happens at a radius of 0.48, and oops, it's going to be cut off a little bit. And then at some point, you increase the radius, and at some point that loop gets filled in, and that would be the death of the feature. So the birth of the loop happens at a radius of 0.48, the death happens at a radius of 0.92, and the persistence, right, so the persistent homology, the persistence of that feature is the difference between the 2. So you could imagine that if you have a really big loop and points kind of finely sampled around it, that feature might last a long time. But if you have a very small loop, then it might not last as long. It depends on a lot of things, of course. Okay, so we have the birth time and death time of features of homological group generators. And so let's suppose this is our data set. We can see that we have these 3 loops, and then there's some noise around it. For all the different features that appear and disappear within our filtration, again, that's where we're growing these balls, it's a Ritz filtration, we'll have the birth time and the death time. And so the birth time and death time is plotted in this persistence diagram. The black circles are for the connected components, the 0-dimensional homology group generators, and then the red triangles are the loops that form and then die off. Features that are far from the diagonal are often considered topological signal, while the features that are close to the diagonal might be considered topological noise. That's not at all how you always want to define it, but it's a common interpretation. You can also, instead of using a persistence diagram, use what's called a barcode plot. And it's the same information. The birth time is the beginning of the barcode, the death time is the end of the barcode, and then you can see its persistence based on the length of the barcode. Yes?
Question. So are the points uniformly selected over the data set? These points from which you start growing?
Oh, yeah. In this case, all the points are used. So every point here actually is a connected component. So they actually all start at time 0 in this data set, and then they start to die off as they become connected. Sometimes you have data that changes with time, so it's not always going to have a birth time of 0. But in this case, they all work present.
In the middle picture, the 3 red triangles, which are further away from, these are the 3 big circles, right?
That is a great question, right? Because we have these 3 big ones, but we also have these.
Yeah, right.
How can you tell the difference? Well, so the problem with using the Ritz complex is that it's not very robust to noise, right? So we can see that we have these circles, but then a circle's also likely going to form here. One could potentially form here, maybe here. Like, so there are all these other places that it could form. So the very last slide before my concluding remarks, I'm going to briefly say how a way of going from the persistence diagram to get a representation of the data, but it's actually not a straightforward thing to do because these are homology group generators, they're equivalence classes, there are a lot of different representations you can find in the data set, but what we would like to say is, well, we have these 3 clear loops, that must be something like these 3, or among the 3 that, the 6 that are kind of far away.
So short answer, you can do that somehow.
You can find, so
You could.
yes.
do that. Yes.
We can find a representation in the data set.
Somewhere.
There may be debate as to how you find that representation, like what is the best representation. But yes, there are ways of doing that, yeah. And in some ways that's like even more important because a lot of scientists want to see what's going on in the data set. So yeah, that's great. So that was with the Ritz filtration, and as I just noted, it's not very robust to outliers and noise, but there are other ways of computing the filtration that is, or at least can be more robust, using a kernel density estimate or something called a distance to a measure function, where basically you do something like smoothing over the data set to kind of wash out the effect of outliers, and so that can be helpful. So just to show you how you would take how the filtration would be constructed when you actually have a function instead of a point cloud. So I'm no longer growing balls, but instead I have some sort of function, and this is what we're going to do with the image of the fibrin data. We're going to look at the intensity of the image and start the filtration parameter at either the top or bottom. In some ways it doesn't matter, but you start the filtration parameter, and as you lower it, you're looking at the upper level sets or the lower level sets of the function, and then constructing the filtration over the upper lower level sets. So at this point, this is the upper level set, and so this is indicating one connected component. as we continue to decrease the filtration parameter, right? So instead of growing the walls, we're now decreasing a filtration parameter. So we have 2 connected components. And then you continue, at some point those merge into one. So one of those 2 died. And then another one over here was born. And so if you're looking at a 2D image instead, you can also get loops. If you do this over a 3D function, you can get voids and that sort of thing. But,
Which one dies?
I'm sorry, what's that?
Which one dies?
So usually they follow what's called the Elder's rule. So in this case, because this one was born first, this one would join that one. So then this one dies when they merge.
The small one.
The smaller one does. But when you have, when you're doing like the rips, it's basically whichever, because they all are born, all the connected components are born at the same time. But yeah, so, but the general algorithmic rule that's used is this Elder's rule. Okay, so there's, I'm going to be using a distance to a measure function. And without going into many of the details, let me just say roughly what this is doing. It's, so we have a point cloud of data, and let's say we want to estimate what we're going to refer to as the DTM function at that point. You have this one parameter that you have to tune and select that's like a nearest neighbor parameter. And so if you want to estimate the function at a point, you're going to find, let's say, the k nearest neighbors of that point and estimate the distance to all the points in the data set, and then just take an average. So what this is actually doing is kind of robustifying the distance function, where you would just say, you know, what's the nearest neighbor, and whatever that distance is what's used. Instead, it's using an average. So it's kind of, in some ways, smoothing over the distance function. And the distance function has some equivalencies to the Ritz filtration. So the idea with this distance to a measure function is it reduces the impact of outliers. And so if we just look at this data set again, this is with the distance to a measure function. So we see that the lower distances, the bluer regions, are where it's closer to the data set. And then the redder regions are have higher average distances, and that's away from the data set. And so this is our DTM function, the coloring is. And then we also have a kernel density estimate, which is where you put down, in this case, you just put down a Gaussian kernel and do kind of a weighted average of the points. And so in this case, the kernel density estimate is higher near where there's more points, so that's why it's redder in this case. But the real takeaway is, if we look at the corresponding persistence diagram, so this is with the Ritz complex, this is with DTM, this is with the kernel density estimate, we see that, as was pointed out, you know, which 3 do we think would correspond to the 3 main loops? Well, with the DTM, you have these 3 that are clearly separated, and so at least the expectation would be that it corresponds to the 3 clear loops. And then also with the kernel density estimate, it cleans up the noise around the diagonal, and so those 3 seem to be well separated. Okay, so doing things like smoothing or a distance to a measure function is can help to reduce the noise. Yeah.
In terms of birth and death for the DTM KD, is it you're varying the width of the Gaussian and seeing when features appear? What are you actually varying?
Okay. Yeah, so in both of these cases, we're looking at level sets. In this case, we're looking at upper level sets. So just imagine a plane decreasing. And as the plane decreases, connected components start popping up. And then at some level, we're going to see loops. And with the DTM, we actually do lower level sets and raise them up. And then you'll see the birth of certain features.
So it's the amplitude of the KD that you're doing.
Exactly, yeah, the amplitude is what's defining in that case, the upper level sets. Exactly. Okay, so what do we want to do now with the persistence diagrams? Well, we want to use them to develop test statistics to carry out hypothesis tests. And, as I mentioned, we're going to look at human versus monkey fibrin. And so these are the 2 images. I just have a note here that we do just a tiny bit of smoothing using local quadratic regression. it was only done to reduce the contrast between the dark and light regions. We actually didn't have to, but it just made the persistence diagrams look less choppy. And I don't actually, I should test this, I have not investigated. I have not used the choppier versions in what I'll explain later. So I don't think it affects the overall performance. It might, I have to check it. But the smoothing, it doesn't actually affect things too much in terms of the structure that we see. Okay, so these are the two persistence diagrams, human and monkey. The H0 are the connected components, and then the red triangles are the loops that are present. And at first glance, they look very similar. But, so we see that we have, so the birth and the death of the loops and the loops and the connected components, they seem to transition around the same spot. And so our question is, can we actually detect differences between the persistence diagrams, suggesting that we could detect differences between the homological structure of the two images. So rather than randomly sampling the different fibrin strands and then measuring their width, that could be time consuming. We can just say, give me those images, let's see if we can find these structural differences. Okay, so the setup is for the hypothesis testing. So we have two sets of persistence diagrams, and they're each coming from some true distribution of persistence diagrams for the different groups. Then our question is that we're trying to test is the null hypothesis that those two population distributions of persistence diagrams are the same or they're not the same. Of course, you can modify it depending on what you're trying to investigate. Yeah.
What are we calling persistence diagram? A single point or a collection of points?
Yes. So actually, it's the part of the slide that got cut off, I think that I defined the persistence diagrams there, which you guys didn't see. This is one persistence diagram. So the persistence diagram, so actually I should modify it slightly. Really what we're going to be testing are the H0 features between the two different samples. I'll explain how I turn them into samples, or the H1 features. So the persistence diagrams are the birth-death pairs and the corresponding dimension. And then actually for the persistence diagram, this gets into maybe too many details, but you assume that there are an infinite number of points along the diagonal. It just makes it so that the theory.
So it's a sampling of two densities?
So in this case, we have two different images. And
No, if you think of this back, so this is two densities in the upper half triangle.
Yes, you could think of it, yeah. So I think of it as like this, more discreetly, I guess, but yes, yeah, that's the idea, yeah.
But when you say that you have n persistence diagram, you assume that you have n images.
You have n images, exactly. You'd have to get n different persistence diagrams. And so that's why being stuck with just one image of each is a problem, and we divide it up, which isn't ideal for a number of reasons. One being that now each sample is correlated, and we don't account for that. But it was more just to set up the method, and then with the hope that one day we could get more samples. Someone somewhere probably does have many samples of fibrin. We weren't able to get access yet. Okay, so yes, this is what we want to test. Then the question is what to use for the test statistic. It turns out that persistence diagrams are actually very complicated objects to work with. The randomness is coming from the space of the data, it's going through this persistent homology filter, and suddenly you have this persistence diagram, and you don't actually know how the features on the diagram relate to each other, right? They might be near each other in the image and spatially correlated in that way, but be very far apart on the diagram, or vice versa. Also, the persistence diagrams, there are distances between persistence diagrams, but they're very computationally intensive, and so you don't necessarily want to carry out that computation multiple times, depending on how one carries out the hypothesis test. But so instead, we want to use something that's a bit easier to work with, and actually it's far more natural for statisticians to work with functional data than with these kind of obscure persistence diagrams. So there have been a lot of there have been a number of functional summaries that take you from the space of persistence diagrams to the space of these functional summaries. And these functional summaries end up being much easier to work with. And for those familiar with statistics, there's a whole field, functional data analysis, where a lot of methods have been developed. And so this goal of, so that's part of the motivation for wanting to turn things into functions. So I'm going to talk about some of the functional summaries. A common one is called the landscape function, and that's what I'll talk about in more detail. But yeah, we're going from the space of persistence diagrams to the space of these functional summaries. And then, so that's saying that our random diagrams, which are random because of the data, they're now going to become random functions that we're going to work with. So each random function corresponds to one persistence diagram, which corresponds to one image. Okay, so given these two sets of persistence diagrams, so we can consider things like the average functional summary, and it turns out that you can show that this average functional summary can converge to the true population average functional summary, which is why we can even think of using these functional summaries as test statistics in our hypothesis testing setting. Okay, so once we have confirmed that we can use those, now we can start defining test statistics to compare the samples of our persistence diagrams via the functions.
So just to clarify, those are IID you received.
Yes, it's assumed here that they're IID, yes. Which does not hold for the fiber data, or actually the cosmology data, unless we had independent realizations. But we do use it anyway, as an illustration. We're not trying to make any scientific claim yet, maybe in a couple years. Okay, so one way to get a functional summary is, so this was our persistence diagram. The idea of the landscape function is to, so you draw isosceles right triangles around each point, and then you rotate the diagram 45 degrees. And then the landscape function is actually a collection of functions that are obtained by first going through your space and taking the maximum, kind of the point-wise maximum, then ignoring that, and then going to the next maximum, and then getting rid of that, and then to the next maximum. So you have these layers that are embedded within the layer above it. And that collection ends up being the landscape functions. It's actually very kind of, I found it very weird, at least the first time that I encountered these landscape functions, but they end up being very nice functional summaries. And rather than thinking of them as layers, you can also instead concatenate them so you have like one long functional summary instead of these discrete layers of them.
So when you do this, you somehow